Welcome to the last series of the training. This training will be focusing on bioinformatics. It is a follow-up or part of the series that we have been delivering through the webinars that we have been conducting. So I will start by introducing the things that we are going to do and the information that we need in order for us to successfully conduct and be part of this training. So we have the Google Drive folder for this particular training. So we are going to access the Google Drive folder, and we should be able to download the files that we need for today. So that is what I'm going to do. I'm going to put the link to the folder in the chat, and then I will show the different files that we are going to use before we can start. And then after that, we will do a pre-survey just so we understand our expectations as regards this particular webinar for this week. So the link to the Google Drive is on the chat. The link to the Google Drive is on the chat. Let's all click there and go to the Google Drive. So I'm going to share my screen to show the Google Drive and what we need from the Google Drive. Okay, so I'm sharing my screen right now. And what you're seeing is the Google Drive that contains the materials that we are going to use starting today. If you can see the Google Drive, type 1. Type 1 for yes. If you can see the Google Drive, type 1 for yes. All right, that's great. So you can also use the chat to ask questions. You can use the Q&A as well to ask questions as we go along. We shall try to attend to the many inquiries that we shall be seeing. So we have 4 items and files that have been uploaded into the Google Drive. So we would like you to download all of them. Would like you to download all of them. We shall use them. We shall use each and every one of them later during the session, but also we shall show you the ones that will be used at a later date. So somebody is asking for the link again. The link is right there. So download metagenomics packages. This will be an assignment that we shall do much later. We are going to use R and R Studio as we've done in the previous training. So download the installation for R and R Studio if you haven't already installed R and R Studio. And then also the Bioinformatics online tools and links. These contain important links that we are going to use throughout this training. And then the activities themselves. The document will contain the activities that we are going to participate in during the webinar series. So it's important that you are able to download all of them, download all these 4 files. And then we shall create our bioinformatics folder. I will show you exactly how to create the folder like we've done before. So if you've successfully downloaded all these 4 files, please let us know. Let me know. We will give ourselves quite a bit of time so that we can access them. Okay, so the link to the drive is there. The link to the drive is there. Access the Google Drive. Make sure you can access the Google Drive because we are going to be updating it quite regularly as we go along. And we shall also let you know if we add any document that you need during the sessions. Okay, so if you have downloaded the 4 files, the other step is for those who have not yet installed R and R Studio. We need R and R Studio. So you're going to open the installation document, this one in particular, and open install R. So there's a link there that will install R, and then there is a link that will install R Studio. There is a link that will install R, and there is a link that will install R Studio. Okay, so okay, so let's check in the chat how many people already have R and R Studio. 1 for yes, 0 for no. Just to get a feel of how many people have R and R Studio. Okay, I see that most of us have R and R Studio. Okay, there are people who don't have it as yet. If you don't have R Studio or R or both, please open the file, this one in particular. So just to guide us a bit, it will be important because for bioinformatics, we are going to use R and R Studio, the bioconductor packages, but also we will use online. So it's important that you have R. So when you open the install R and R Studio document, you will find the link for downloading R and R Studio. You go to CRAN and you'll find the link to install R. You go to this other one and you will find the link that installs and downloads R Studio on your PC, and so that you can run that installation very fast. All right, so now we are going to
Please,
move.
can you share the Google Drive? Somebody's, I think, asking
Okay,
for.
all right. So the link to the Google Drive is there. The link to the Google Drive is there. So for those that haven't installed R and R Studio, please go ahead and click on the links that are in there. If you're using Windows, choose the link for the Windows, and we proceed. All right, so once again, somebody is asking on the guidance for the installation. If you open the document, so we are doing this because we will need R and R Studio. Now, if you open the document, if you're using a Windows PC, install R, click on this link here, okay? And then you should be following those instructions to install R. And then after installing R, you click on the link down here to install R Studio as well. So you will need to install both R and R Studio because R Studio will be the user interface that we shall use. If you are not using your Windows PC, if you're using Mac, then install R using this link here. And then after, install R Studio using the link that is stipulated for those who are using the Mac operating system. So we all have this document in the Google Drive. Please use it to do an installation for R and R Studio. So the only thing we shall use R and R Studio for today is to create the bioinformatics folder. The actual using of R will be done tomorrow. Okay, will be done tomorrow. And we will have our assignments on how to install the necessary packages. You will go and do that later after this course so that tomorrow we have the necessary packages that we need within the R workspace. All right, so that is something that you can continue doing. Now, the other thing that we will request you to participate in now is the pre-survey. So we are going to take a few minutes to participate in the pre-survey so that we can understand where we are at and we can be guided in what skill sets we need to attend to during this webinar. So the link to the pre-survey is there. Please click on the link and fill in the survey. It should take a few minutes, but we are going to give ourselves some ample time so that we can have as much information as we can. So the link to the survey is shared on the chat. Please click on the link and access the form. Click on the link, access the form. And then submit when you're done. And then submit when you're done. Yes, and if you're done, you can let us know in the chat. So somebody has a challenge, an error when downloading the files. Are you able to access the Google Drive itself? Are you able to access the Google Drive? And what error are you getting when you're trying to download the files? You need stable internet in order to be able to download the files. You need stable internet in order to be able to download the files. The survey link is here. The survey link is right there. Please continue to fill in the pre-survey. And those that are installing R, please continue to download the installation file. They're not very big, so it should take a pretty short time to download and install. Also, make sure you have permissions to install software on your machine. Okay, so we have a few more minutes to fill in the survey. A few more minutes to continue filling in the survey. Somebody is asking, what are we installing? For those who don't have R and R Studio, you are installing R and R Studio. For those who don't have R and R Studio, the Google link, the Google Drive link is here. The Google Drive link is there. That's the Google Drive link. As you join, please make sure you're accessing the link. And in the Google Drive, please just download all the folders that are in, rather all the files that are in there. And also ensure that you fill in the pre-survey. The link for the pre-survey is also there. All right. Okay. The link for the survey. The link for the survey is there. Somebody's asking, how do you advise on using AI tools to develop an R script? Okay, an answer is coming your way. An answer is coming your way. So if you've accessed the Google Link Drive and you have submitted the pre-survey, Please type 1 and then we can start. Type 1 and then we can start. Okay. We are waiting for a few more responses. Okay, I see that we have successfully submitted the pre-survey. So the link to the pre-survey is here. In case you missed it, I have posted it again. The link to the pre-survey is there. In case you missed it. All right, somebody's saying wait for 2 minutes. Okay. So in the meantime, as we wait for you to submit, okay, so somebody said they're done with the survey. What tools do we need to install? No. From the list, we only are installing R and R Studio. R and R Studio, that is what we need to install. Any other package that we need to install will be given later, guidance will be given, and we will do it together. So for now, you need to install R and R Studio. That is the tool. So the PDF document that is inside the Google Drive that contains R_R install is what you're going to open to install R and R Studio if you don't have it as yet. If you don't have it as yet. Okay, so thank you for everybody that has participated in the pre-survey. Now we are going to, okay, so we are going to create a folder for bioinformatics, and then we will kickstart the training. So I'm going to share my screen. I will take you through creating a project that will create a folder. And then after, we will start the training. So right now, I am sharing my screen and I am sharing a PowerPoint presentation. Hopefully, let me see. Okay, so I am sharing a PowerPoint presentation. Okay, in the chat, confirm if you can see the presentation. 1 for yes. Okay. All right. That's great. So welcome once again. We are going to start off by introducing the tool itself that we are going to use, and then we create our project, and then we start the presentation. So we are going to be looking at bioinformatics for research and institutional development for the next 5 days, including today. So what are we going to cover in this particular week? We are going to look at what bioinformatics is in detail, what it means, what it entails, what you need to know, okay? Especially the prerequisites, what to expect as well. Yeah, so after that, we will look at sequence analysis. Then we shall also go into metagenomics. We shall look at transcriptomics, that is the RNA-seq, and then we will conclude the week by looking at genomics. So this is what we expect to cover this week. So the week is going to be intensive. The week is going to be intensive, but we will be guided through using both the online tools and also R and R Studio, the different packages in Bioconductor that are going to help us to navigate through the different assignments and to understand the different concepts that we are going to look at throughout the webinar. So because this is going to be more of a hands-on, we are going to create a project. Now, in R, it's important that you have this project created, because all the material that we are going to put in the Google Drive folder will need to be downloaded and put in this folder so that we do not get challenges in dealing with our working directories, especially for this particular training. All right, so in R, if you've installed R and R Studio, then you are going to open R Studio itself. You're going to open R Studio itself. You should be able to see these 4 windows. I will do that practically. If you're not able to see them, we shall see how you can be able to see them. But most importantly, you should make sure that you have opened R Studio and not R itself, okay? Because R Studio will give us the user interface that we want to use that is easy for us to use to navigate R. So in creating the project for our bioinformatics training, go to File and click on the new project. You will find the new project under File. And then we'll go to a new directory. Okay, so because we
Please,
want
somebody
to,
has only 3 windows. Can you help him?
yeah,
Somebody have only 3 windows.
okay, somebody has only 3 windows. Yes, I
Yes.
am going to um, go there. So let me first go into RStudio and address that. Then we shall come back and proceed from here. So when you open RStudio, you can have 3 windows, okay? You can have 4 windows, you can have 3 windows, but if you have 3 windows, you can display the other window from here. Okay, you see these 2 windows here, you can split them so you can have both windows. Or you can go to this icon here, workspace pens, and show all the pens. And then you should be able to see all the 4 pens. Before we come here, this window here that you're seeing will be our script editor. We shall navigate and use it together. Here we have the environment where we are going to have the different data that we are going to use. Down here, we have the R console where the execution of the codes that we do are going to take place. And then on the lower here, we have the files, the working directory. We shall have the plots. We shall see them. We shall also use it to install some packages if we are not using an R script to install the packages. So for starters, your R Studio should look like this, okay? You should be able to have all the 4 windows. If you have 3, you can always display them from there, or you can go to the pens and just show all of them, and so that you can be able to see all of them. Let me see if I have a. Okay, so I'm going to proceed. So I have one demonstration. For R Studio. So somebody's saying those who are installing, it's fine. Please continue to install. The aim today is actually to install R. For those who haven't installed, our main aim is to install R and R Studio and create a project where we are going to put all our information, and then we will start using R tomorrow for any analysis. Okay, so we are going to create that project, and we create a new project in a new directory. Especially, we want you to put it on the desktop, and then we should be able to see the project. We are going to name it Bioinformatics, and then after, we should be able to see the folder on our desktops. Okay, so what I would like us to do is what I've shown you. I would like us to create a folder called Bioinformatics, where we are going to put all the things that we download. So normally we do that practically, and that is what we are going to do in RStudio. As we give time for other people, create a Bioinformatics folder, go to File, click on File here, go to a new project. Under File, go to New Project. And then go to a new directory. So we are going to create it in a new directory, and it's a new project. Okay. So make sure that your project is on the desktop. You can always browse from here and go to your desktop wherever it is and open it. And then give it a name, Bioinformatics. And then once you're here, you're going to click on Create Project. Before I even proceed to create the project, let me pause. Let me pause and find out if you have reached this stage. Let's find out if you have reached this stage of creating the project. And if you've reached this stage, click on Create Project. Click on Create Project. All right, so I see people have reached this stage. Click on Create Project. Somebody says we should repeat. Okay. And when you create, yeah, you put the documents that you've downloaded from the Google Drive, you're going to put them in there. Okay, so somebody says we repeat. So I'm going to.
Please, can you repeat having 3 to 4 windows? I think some people still have that problem.
Okay, so we are going to repeat. So I'm going to go back to RStudio. So right now I'm on RStudio. If you don't, somebody's asking, is this R you are recommending us different from the one we have been using? No, it's the same. We're using the same R and the same RStudio. If you have it, then yeah, okay. So now, if you have only 3 windows, which is probably something like this, you can display the windows by going to this workspace panes that is up here. These 4 windows here, 1, 2, 3, 4. You click on those and then click on Show All Panes. It should be able to show you all of them. It should be able to show you all the 4. You can adjust the size, okay, by placing your cursor at the intersection of any of the 2 windows. Okay, so you can navigate and have them, and you can adjust the size. You can adjust this one. The aim is to have all the 4 windows here. Okay, so we are now going to create the folder again. It's important that you have the folder. Now we are creating a folder for the bioinformatics training. Previously, we have created folders for different trainings. So come to File, go to New Project, go to New Project, and go to, no, we are not creating another folder again. For those that have not yet created the folder, I am repeating the step so that we all have the folder. If you've created the bioinformatics folder, you don't need to create it again. Okay, so go to a new directory, and then new project. So we are creating a new project, and make sure that you browse and put your project on your desktop. So find where your desktop is and open, so that in the directory down here, you are going to create the project on the desktop. The name of the project is Bioinformatics. Okay? And then click on create. So when we create the project, again, you might have only 3 windows, like I do now. After creating the project, I might come up with only 3 windows. You can just click on these 4 panes here so that you can display all of them and adjust them accordingly. That is one. Also, when you have created the project, it should display here the bioinformatics project, because right now that becomes our working directory. The other thing is that if you're working within the bioinformatics project, then you will see the name of the project up here, which is bioinformatics, but we are in our studio. So this is what you need to have as of now. So, okay. Somebody's asking for the link to the Google Drive. Let me know with the one there if you have successfully created the folder. If you have successfully created the folder, one for yes, one for yes. Okay, that is great. So now when you go to your desktop itself, you should be able to see the folder. Mine is here. You should be able to see the folder on your desktop. And when you download your items from the Google Drive, you can paste them inside this folder. This folder will be important for all of us. So we can continue. Since we have all this, I'm going to stop sharing my screen and I will hand over to Stephen. Stephen, you can proceed.
Thank you very much, Peace. So now we are going to start the webinar for today. Share my screen here. If you see my screen, type 1. Okay. So I'm going to start from where Peace stopped. So what's bioinformatics? We have covered the last few models. We talked about data management. We talked about data science. And our last model is bioinformatics. So what's bioinformatics? So bioinformatics is a discipline that combines biology, computer science, mathematics, AI, generative AI, statistics, to analyze, interpret, and visualize biological data. So the key here is biological data. When we covered data science 2 weeks ago, was any kind of data. But in bioinformatics, we're going to use almost the same tools that we use in data science, but you are restricted to biological data. And we will see those biological data later. So a few decades ago, when people started bioinformatics, that was introduced maybe 2 or 3 decades ago, it was basically tools. But now bioinformatics is driving discovery. So what does it do? It enables researchers to analyze vast amount of data that's generated by sequencing machines when you're dealing with metagenomics, transcriptomics, and genomics. And then data that's generated by mass spectrometer when you're dealing with metabolomics, imaging platforms, even GIS and monitoring like drones, satellites, and field data. So any data that's biological, when you're analyzing it, interpreting it, and visualizing it, you're dealing with bioinformatics. So sometimes I'm asked, is bioinformatics also data science? Bioinformatics is under data science, but bioinformatics is not data science. Because data science, you can also analyze biological data. You can analyze any type of data. But bioinformatics, you're only restricted to biological data. That's the difference between data science and bioinformatics. But the tools are the same. You need computer science, you need mathematics, you need AI, and so forth. So how do you work with bioinformatics? The first thing you have to do is collect data. So the data is the raw materials, for example, like DNA or RNA. So for collecting your raw materials, you have to check the quality of the data. It's like what we saw in data science. You collect your data, you check the quality to make sure that it's clean and it's reliable. So if it's not reliable, you discard those ones that you think there are problems. So if you match those data, putting them together like solving puzzle, you label those data to make sure that maybe if you have a genes or mutation, you analyze those data to find meaningful results, and sometimes you visualize them. Because at the end of the day, if you collect the data and you don't get the results that's going to give any important meaning, then there's no use of doing bioinformatics. So you have this process. You collect the data, you check the quality of the data, organize the data, you label, analyze, and you visualize. Okay, so when we covered data science 2 weeks ago, we talked about the core of data science. We have descriptive analytics, diagnostics analytics, predictive analytics, predictive analytics. But in bioinformatics, we also have, so the core of bioinformatics is called omics. So omics, we have different types of omics. We have genomics. So genomics basically focuses on DNA sequence. So this includes identifying genes, but we are going to cover that in details, maybe on Thursday or Friday. Mutations, the structural variation. And then we also have transcriptomic, and some people call it RNA-seq. So this shows the expression levels whether genes are turned on or off. So this by itself is a field that is in bioinformatics. It's core of bioinformatics. And then proteomics. So this proteomics basically investigates the structure and function of proteins. And the proteins are the true working horses of the cells. So the next few slides, we'll see how proteins are made, but they're the true working horse of the cell. So genomics, that focuses on DNA. Transcriptomics, that explore how this DNA is turned on and off. And then proteomics, and investigate the structure and function of those proteins. And then metagenomics. So metagenomics basically analyze the genetic materials from the environment. For example, you have clinical samples or other things from basically deal with microbial communities. So when you're doing metagenomics, which we're going to cover, I think maybe tomorrow, always dealing with that culturing bacteria or fungi. And then you have metabolomics. So metabolomics, basically, you're profiling small molecules basically it is the end product of omics. So you start from genomics, the DNA, and it transcribes to RNA, and then you have your protein, and the protein, the byproduct, you have your metabolomics. And then last is phenomics. Basically, this is what you see, the traits that you see, the morphology, physiology, and so forth. So those are the core and the scope of bioinformatics. That's why they're called omics, okay? Like what we covered in data science. So in bioinformatics, we have these omics, and each of them are different, and each of them have the way of analyzing them. They have the specific niche in bioinformatics. Okay, so why do you study bioinformatics? Because bioinformatics has a core function. So first is data management. Bioinformatics help us to store, organize huge amount of biological data so that scientists can be able to find those data. Okay, so we'll talk about databases. So for example, if you do your experiment, you do your experiment, you sequence and you publish, you like people to use those sequences in the future. So you submit the sequences to a public database. So they help to store that. Another core function is sequence alignment. So this compares different DNA or protein sequences to see how similar they are. And one of our activities here is going to be sequence alignment. So it's help in identifying patterns, maybe in mutations. It also helps in seeing how evolutionary has taken place, because if you align these sequences together, similar proteins or DNA will be aligned compared to the ones that are not similar. Another core function of bioinformatics is gene annotation. So what do we mean by gene annotation? Basically, this is labeling DNA and showing which section of genes, what they might do. Okay? And also we have structure prediction. Bioinformatics helps in predicting structure of the protein, because structure of the protein, it has been the toughest part of bioinformatics to do, because used to be you do experiments, sometimes take years to predict only one structure of protein experimentally. But in the last few years, bioinformatics has done a very, very good job that you can now at least predict a structure of protein with between 90 to 95% accuracy, that by predicting that if they go and do experiment, the structure will be very, very close. So that it has helped in bioinformatics. So 2 years ago, the Nobel Prize was won by people who broke the code of predicting protein structure. But that has been the biggest, the toughest part of biology, predicting 3D structure. So bioinformatics has helped in doing that. And then functional analysis, it helps to figure out what genes and protein are actually doing in the body, like their role in diseases. So bioinformatics help in doing that. Okay. Okay. In order to do all this, we need tools, because if you don't have the tools in bioinformatics, we'll not be able to predict functions, to store all these sequences. So the most common tools that are used in bioinformatics, the first one is called basic local alignment search tool. So anybody who works in bioinformatics should know this tool. It's called BLAST. So BLAST, what it does, it helps scientists to compare DNA or protein sequences with millions of others in the database. One of our activities is going to be this one. We're going to use BLAST. So assume you are a researcher, you got your sample, you extracted DNA and sent it for sequencing. After sequencing, it was brought back, and you don't know what that DNA is. The first thing you always do is do the blast. You blast search, and then it will bring for you similar DNA or proteins, and then you infer from that okay, what I've sequenced from my experiment is similar to this sequence, either from bacteria or from other organisms. Another tool that I commonly use is Galaxy. So Galaxy has solved a lot of problems, because sometimes people used to analyze their data on their computer. But Galaxy has made it easier now. You can do it on the cloud or can use it on high performance computer. Then RN bioconductor that we're going to use in the next 5 days. It has helped a lot in bioinformatics because of the packages that they have. So ARA and bioconductor have now tools for bioinformatics that you can use in any of the omics, whether using when you're working with metagenomics, if they're working with proteomics, whether they're working with genomics, they have packages for that. So the next few days we'll basically go through, that's what we'll be using. And then also Python. Python has very many libraries that can help in bioinformatics, but we're not going to use Python here. Let me check if somebody has any question here. Okay. Biological data and databases. Okay. Okay. How do you get from sample to sequences? Because somebody might be asking, okay, we've been talking about these sequences. How do you get these sequences? So sequences always come from researchers. So what they do when they do experiments, they collect their sample, they take small amount of materials like from saliva, plant liver bacteria. They extract the genetic material, it can be DNA, RNA, and pull it and purify them. And then this, of course, is broken down and then sent for sequencing. Then after sequencing, they're sent back to the experimenter. And then from there, that's when now you start doing some bioinformatics analysis. But before that, you have to get the sequence. So how do you get it? You get your sample, extract either DNA or RNA, you send it to be sequenced, and then they send it back to you. So basically, this is like a cartoon. You collect sample, extract your genetic material, send it to the sequencing, and then they send it back to you as a in form of sequences. Okay. So in order to keep the sequences that maybe you're an experimenter, your sequence, you can keep on your computer. Some people keep that on computer. But these days, when you're going to publish, one of the requirements is that you have to deposit your sequence in some of these databases. The most common one is a GenBank. GenBank is basically its own NCBI. Stores DNA from scientists and put it all over the world. And it has other things. So when you go to GenBank and you get a sequence, it will give you sometimes information of it. The person who did the experiment, when the experiment was done, and so forth. So this is one of the most important databases that I use. And then another one is UniProt. UniProt is based in Europe. This basically keeps protein sequences, and also it has other things that help people. A person researcher, we'll use UniProt when we are doing RNA-seq. Then last but not least is Protein Data Bank. So this stores all the 3D structures of the protein that has been determined experimentally. So when you have your protein, you have your protein, and you want to predict the structure, you can use the 3D, the PDB. So these are the most common databases that are used in bioinformatics. There are others also out there, but these three are the most common databases that are used in bioinformatics. So when you're doing BLAST, basically you're using GenBank. And when you're searching for proteins, sometimes when you're using for motif and so forth, you're using UniProt. And when you're searching for protein 3D structure, you're using a protein data bank. Okay, so what I'm going to do now is, before we continue, I would like us to watch a small video here to show something about how do you get from DNA to protein, because that is going to guide us for the rest of the webinar today. So let me go to this video here.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay. So what we've just watched, can you see if you see my PowerPoint, just type 1. Okay, thank you. So what we have just watched is called central dogma of molecular biology. So in the nucleus, we have this DNA, okay? And then before the transcription takes place, the introns is removed. And then after that, it's exported out of the nucleus to form amino acids and then into protein. So this is called the central dogma of molecular biology. That's what we have just watched. So I wanted us to watch this because from now on, we're going to go the bioinformatics part of it. To how do we use tools so that we can do the same thing like what we just watched? Because as I mentioned before, that assuming you're an experimenter, you got your sample, maybe you went to the field, you have a plant that was diseased, you pick that plant, you extracted your sample, extracted DNA, send it for sequencing, and then you come back, they send you back the DNA. So what do you do from there? So from now on, you're going to see how do you work with those sequences, okay? So before we do that, I would like to at least go in detail and talk about this biological type data. What are the types of biological data? Okay. We'll start with DNA and RNA. So DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Both of them are nucleic acids, by the way, and that carry genetic information, but they have distinct structures and functions. So the structure of DNA is double-stranded helix with sugar phosphate, and the bases are ATGC, and the sugar is deoxyribose, lacking one oxygen. But RNA is single-stranded, and the bases are AUGC, with sugar with ribose instead of thymine. Okay, that's structurally DNA is double-stranded, RNA is single-stranded. Okay. How about the location? DNA is found mostly in the nucleus, for example, in eukaryotic cells, and also in mitochondria and in chloroplast. And RNA is found in both nuclide and cytoplasm. What are their functions? DNA stores and transmits genetic information. This is the blueprints for proteins. You know, when I showed you where it started from DNA to protein, start from transcription and translation, so it's the blueprint for the protein. But RNA, there are three types, okay? There's messenger RNA that carries DNA codes to the ribosome. Remember when you watch the video? So after getting out of the nucleus, it went out and carried that information that it was transcribed from the nucleus. And then we had also transfer RNA that was bringing those amino acids. Okay? And then we have ribosomal RNA that's basically for structure for the ribosome. So for genetics, for carrying information, we have messenger RNA that carries the information from the nucleus and brings it to the cytoplasm, and then the transfer RNA now brings the amino acids. Okay. So this is just a cartoon for DNA. It's double-stranded, and RNA is single-stranded. And the sugar here, thymine, is represented by uracil and so forth. So again, DNA is transcribed to messenger RNA transcription, location nucleus in plant and animal, process a gene, the DNA segment for the protein. So it's copied the messenger RNA, and the enzyme is RNA polymerase. So the results, it carries the instruction, the genetic code out of the nucleus to the ribosome. So that's the function of messenger RNA. Because DNA is basically in the nucleus, the genetic code has to be carried from the nucleus outside so that it can be converted to the protein. And then messenger RNA now translated to protein. Location, ribosome in the cytoplasm. The process, you read this in codons. Codons basically there are three bases. And each codon corresponds to specific amino acid. We're going to see that how in the next few slides here. So each amino acid has a specific three types of bases. So the transfer RNA will bring those amino acids depending on the type of the bases. And then keep adding until it reach what's called a stop codon. And then it will stop, and then the protein will form. Okay? And then after the protein folding, sometimes they're modified so that they can add sugars, they can add lipid, phosphate, and so forth. So basically, after folding to 3D structure, other activity can also take place. Okay. Somebody has a question here. Let's see here. Okay, so I've talked about DNA. I've talked about RNA. There are 3 types of RNA. Now I'm going to talk about amino acid. So what's an amino acid? So amino acids, these are organic molecules that contain amino group, carboxyl group, and hydrogen atom. The only thing that's unique from each amino acid is called the RH group. So if you look at this is alanine, okay? Somebody has raised their hand here. So this is alanine. The hydrogen for alanine, and this is valine. The hydrogen, the amino group, and carboxyl group are the same. But the only difference between alanine, valine, and isoleucine, and leucine is that these are a chain here. So they're the building block of the protein. Okay? So when we watch the video, it was mentioned that there are 20 different amino acids. So these 20 different amino acids are the ones that the building blocks of protein. And each of them, each amino acid has its main 3 codons. So I'll show you in the next few slides. So that when a transfer RNA comes, it depends on the type of codon, then each of that amino acid will be added to the chain. Okay. Okay. So DNA and protein. So how do you print DNA? How do you represent DNA protein? So DNA is represented by the 4 bases. So for example, this is a DNA of sheeppox virus. So you can see presented by the AT, GA, and so forth. So they are presented by the 4 bases. And the protein are represented by the 20 amino acids. Okay? So these are in the FASTA format, which I will explain later. So when you're presenting DNA in the bioinformatics, you present it in a form of this is a FASTA format. We present them in their bases. And when you're presenting protein, you present them in the amino acid, with the amino acids. Okay? Let me check here if somebody has a question. But it's not for me. Okay. So our first data structure that is very important in bioinformatics is called FASTA format file. So as we go on, we're going to see other data type in bioinformatics. So this one is called FASTA format. So FASTA format has two parts. It has the header and the sequence. So the header always starts with the greater than sign, a symbol, and then gives you information about organism or the accession number. Okay? And the second line are the sequences. So if somebody send you a FASTA file, it will have 2 components. It will have an header that give you information about the sequence, and then after that, it will give you the sequence. Like this is a DNA. It will the sequence will be presented by the 4 bases. Okay. So the structure for protein, again, you have two parts. The first part is the header. We will give information about the protein, the section number, and then the sequences will be presented by amino acids. So when you're dealing with protein, they're presented by the amino acids. And when you're dealing with DNA, they're presented by the nucleotide, the 4 bases. Okay. So this is what I mentioned before that I'll show you. This is what they call genetic code. So when we were watching that video, the transfer RNA was bringing those amino acids and adding them into chain. But each amino acids depends on the codons on the 3 nucleotide. So for example, for amino acids to be aniline, the codons must be one of these. So if the RNA has maybe this one, it will never add amino acid alanine. It will add amino acid arginine. Okay. So this genetic code is very, very important when we are trying to translate our protein, our DNA in the protein. So, and the most important one is the stop codon. So when the stop codon, one of these 3 added, the protein, the amino acid that will stop. That will be the end of adding the amino acids to that protein chain. So these 20 amino acids, each of them have their own codons. So when transfer RNA brings an amino acid, it has to correspond to each amino acid has its own codon. Okay. So in bioinformatics, there's a way that we can do this. We have tools for doing that. But to do that, we have to use our genetic code. So assume this is your DNA sequence that you submit. You did an experiment. You send your sample for sequencing. You got this DNA. Okay. Now you don't want to know what from this DNA we want to predict your protein. So the machine has to know when they're translating this, the machine will use a genetic code to translate that. So when you're translating your DNA to protein, it will use the genetic code that I showed you from the previous slide to do that. Okay. Okay. So this now brings me to what's called 6 frames. Sometimes when in bioinformatics, people talk about 6 frames. I mean, I always check if somebody has a question for me here. I mean, others. Next this one. So if people talk about 6 frames, what does that mean? So I'm going to take my time here and explain what they mean by 6 frames. So let's take this DNA sequence. So it starts from ATG up to TTM. So the codon has to have 3 bases. So you can start translating, you can start bringing amino acids using these 3 bases here, ATG. So this will bring amino acid, and then the next will be AAT, and the next one will be TAT. So this is going to be the frame number 1. Okay, let's repeat again. You can start by using these 3 codon from the beginning. ATG, that's 1 amino acid. AAT, another amino acid. This one's another amino acid, and until you are done. That's the first frame. But you can also start from the second amino acid, the second base here. You can start from T. So TAG, this one will give an amino acid, and then ATT, another amino acid, and so forth. So this is frame number 2. And you can also start from the third one here. So GAA will give you amino acid, and so forth. So we have frame 1, frame 2, and frame 3. You can also start from the end. You can start from here, ATT going backward. This will give you amino acid, ATT, another amino acid, and so forth. That will be frame number 4. You can start another at the second last position, TTA, it's a different amino acid, and so forth. So you have frame 1, 2, 3 up to frame number 6. So you can have the DNA can be translated into 6 frames. That's what they call 6 frames. And out of those 6 frames, it's most likely you can have most of the time you can have only one protein. You can have it either from frame 1, or can be from frame 2, frame 3, or up to frame 6. Okay. So when people talk about 6 frames, when you're translating your protein, your DNA to protein, that's what they mean. You can start from the first position, the second position, and the third, when going from left to right. And you can start from the last position, going from right to left. Okay? So basically, this is what I've explained. So the forward strand has 3 possible readings depending on whether the translation starts. So sometimes when you read your results, sometimes they call plus. The plus ones are the forward strand. So frame 1, the codons are read from base 1 to 3, 4 to 6, and etc. And then often the longest biological relevant OR is the one that is the longest. We'll see that later. And then frame number 2, which is plus 2, start from position 2 to 4 and so forth, and frame number 3 and so forth like that. So that's what they mean by open reading frame 1 to 6. Okay? So the reverse, they're always minus. You start from the last, second last, and the third last. The same thing. Okay? Okay, so how do you decide after translating all the 6 frames, how do you decide which one is a protein? Really is a functional protein. So first, when the translation takes place and the stop codon appear in the middle, then that cannot be a functional protein. And then also, a protein always starts with ATG, that is methionine. And then sometimes what you have to do is to check whether that protein has a constant domain. Okay? But viruses, sometimes they can have 2 or 3 frames, but eukaryotes always have 1. So when you translate those 6 frames, check to make sure that the stop codon not in the middle, and then also starts with methionine. Okay? Okay. So I'm going to demonstrate to you, and then we'll do some activity. We have an online tool that can be used to help you translate your DNA to protein. And it is an emboss, and it's called trans. So when you go there, it tells you the type of sequence that you can use. You can use FASTA format, you can use other format, and then you just paste your sequence here. Another thing that you can do, you can also choose, if you have your sequence in a file format, you can also choose your sequence in a file format, okay? And then it has these different parameters. You can use one frame, or you can use all the different frame, which I'll show you. And then it has the type of codons, the standard one. And then you can give it a title and you submit, okay? So I have a sequence that I'm going to demonstrate to you. So this was the ship virus sequence. I use it. And then the first time I did, I selected only one, frame number 1. Then after submitting, I also got a standard, always use the standard code, okay? After submitting, so I got the sequence. I selected the parameter 1, and the code was standard. And then I put the name Bioinformatics 1 frame, and then I submitted, okay? So after submitting, when it's running, it tells you that the job is running. You see something like this, okay? And then it gave me this. I would like you to watch this. So the first amino acid was methionine, that was okay. Okay. And then the first stop codon was here. The star is your first stop codon. So my protein basically start from here and stop here. Okay. So anything after this is not part of the protein, because basically when we're watching the video, when they're adding amino acid, they reach a stage where it is stopped. So this star here means it has reached a stop codon. That's the stop codon tells the machinery to stop making the protein. So when I'm now trying to, from my sequence, this is my protein. It starts from this and stop here. And that's the protein that I'll be using for maybe doing blast and so forth. Okay. And then also you can see it in color. So if you click on that, it will give you different colors of the amino acid. Okay. And here is the stop codon. So when from the sequence, the DNA sequence that I got, this is the protein. But anything from here onwards that it's not part of the protein. Okay. So that was when I used I selected one frame. Okay. So I went back and did the same, but this time, instead of selecting one frame, I selected all the 6 frames. Okay. Everything was the same except I selected all the 6 frames. Then after submitting, these were my results. So you can see from here, this is frame 1. This was my stop codon stopped here. So this is my protein. If you look at this frame 2, the first position was just stop codon. So basically there's nothing much I can do. So this is not even a protein. So if I go to frame 3, you can see I have stop codons everywhere. If I go to frame 4, still I have stop codons everywhere. If I go to frame 5, the same thing. Sorry. Frame 6 also the same thing. So basically, even if I'd use all those 6 frames, my protein is this one here, the first frame. Okay. Okay. So what are these colors here? Because sometimes when you're using online tools, you find these amino acids with different colors. So these colors have meaning. These colors have meaning. So these colors are basically chemical properties of amino acids. So they can be hydrophobic, polar, charged, or aromatic. So any amino acid that's red It's either charged, it's either acidic or basic. So you have aspartic acid, glutamic acid, lysine, and arginine. All those are what? Indicated red. And then the blue ones are hydrophilic or polar residues. So these are basically asparagine and glutamine. And the magenta, which is speckled pink, these are often discharged. They're kind of lysine and arginine and so forth. So when you look at these colors, they have meaning. So or when in bioinformatics, it will help you to find out if you're doing the alignment of if you're working the protein, which of these physical properties are most common. Okay. Okay, so I'm going to go now and demonstrate. I'm going to go to and demonstrate, and then after that, we're going to do some activity together, okay?
Hello,
Okay.
Steven.
Yes.
You have some questions in the Q&A.
Sure. What's the question? Let me see here. If I, let me see the question here. Oh, somebody asked, okay, I'm going to answer this question. How long does it take to convert DNA to protein? It depends on the size of your, okay, I'm going to answer using the tools because we're going to use online tools. One, it depends on the size of your DNA. And two, it depends on how busy that server is. So if the server is not very busy, will take a short time. But if the server is very busy, will take a long time. But also, if you have a very long protein, it will take time too. Because sometimes when you submit your protein, those servers they rank them based on the size of the protein. So let me look for another question here. Okay, so what's the aim of grouping the amino acids like that? So we are not grouping them. So amino acids are the building block of proteins. So proteins can be enzyme, can be structure. So they're not grouped. They're just, the protein is the result of the gene in the DNA. So from the DNA, that information is brought by messenger RNA. So that information is converted into protein. And the protein, to be protein has to be amino acid. For example, let's take I'm going to talk about agriculture. Let's take maize. So if you have a maize that's infected, okay, and you sequence that bacteria that's infecting that maize. So you send that for sequencing, you get the DNA. So that DNA will be translated to protein. So that protein, this is going to be the order of those amino acids. So this order will do what will when you get that amino acids and you blast it, it will show you what bacteria is that. Okay, so the amino acids are not grouped just randomly. They are information from the nucleus, from the gene. Okay. Look for another question here. So after the six, will it repeat itself? Okay, there are six frames. Six means, let me go back and answer this very well because the question is, after the six, will it repeat itself? Let me go back here. There are only six frames. Let me go back to my PowerPoint. Okay. So why are they six? Because let's start from the beginning here. This is one, the first one. You need three codons to make amino acids. So you start from here. ATG, this is one. AAT, the second one. TAT, the third one. So again, when you start with ACT, it's going to be the first frame. So these three, okay? Now when you go to the second frame, you're going to start with second position. And when you stop here, when you repeat again, will be frame 2. When you start with the third frame, you're going to be at the third position, okay? And when you go and you start repeating. But now when you come to the sixth frame, so this is the fourth one will start at this position here, okay? And the fifth start at the second position. And the sixth start for the third position. And we'll keep repeating. So when this is the frame number 6, okay, this is the third amino acid. And then the fourth will start from here. The fifth will go from there. There's no seventh one. So you have 6. There are only 6. Because if you start from 1, after 3, the fourth one will still be 1. You start from 2, after this, it will also still be 2, and so forth. So there's no 7th frame. Okay? Let me go back to the question again. Okay, that was that. Okay, what are the different type of codes for? So the different type of codes are for amino acids. So let me go back again to my slides. So when we watch the video, the transfer RNA was bringing those amino acids. So those amino acids they are not random. So for amino acids to be alanine, to be brought by transfer RNA to be aniline, the codon has to be either GCT, GCC, GAC, or GCG. So where does this come from? So in the beginning, we have genes in the nucleus. So those genes are made of DNA. So if a gene is switched on, for example, let's think about climate change. So we have a drought, okay? But out of those drought, they have one, out of those, you go into a maize field, but there's one maize that has survived. There should be a reason why that maize survive. That may survive because of genetics. And what's that? What did that genetic? So the genetics came from where? It's the gene. So in that nucleus, there's a gene that's a drought tolerance. So how did it work? So that gene was switched on when it was very hot. So it was switched on. So after switching that gene on, it was transcribed. So it went out, was transcribed, and it made into protein. And that protein protected that maize not to die. So let's go back to the video again. I think that will help. So you watch this, okay?
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so let me go and answer that question now. Okay. So I'm going to go back again to the drought resistance. So you're a breeder, you have this maize field, and didn't rain for a long time, but there's 1 or 2 maize that survive. Why did they survive? So let me just take one. One of the gene for drought for heat was switched on. So when there was no rain completely, that gene was switched on in the DNA. So that gene was switched on and then was transcribed. But after transcription, that gene was converted into protein. And the process was, after switching on, messenger RNA was made, and then the messenger RNA moved out of nucleus to ribosome, and then so now this is now where the genetic codes comes in, because when the gene was switched on, there is an order. It's not random. There's an order. So when those codes were out, so the RNA was bringing those amino acids based on the genetic code. Okay, so the genetic code, so like alanine can only come when it has specific 3 codon. So after that, and then from the protein, that protein now was able to protect that maize so that it survived the drought, so it became tolerant. Okay? So everything comes from the gene. The first switch on in the nucleus, and then it's transcribed, and then the rest is history. Okay, so let me see here. Let's see if I have some questions here. Okay. During transcription, is any information lost? No, I don't think so, because I don't think any information is lost during transcription. Okay, so I think we are okay. I've answered those questions. So let's take a 5 minutes break. Then I'll demonstrate the translation. Okay. Take a 5 minutes break. Meanwhile, I will also keep answering questions. Let me see. Any other question that I can answer, I will be happy to answer. Let's see here. Mhm. Okay, so what I'm going to do, I'm going to demonstrate to you, and then you have activity that you downloaded from the Google Drive, and then we'll do exactly what we've done. So this is type 1, you can see my screen. This is the website. Type 1. Okay. So this is the website. So on the left here, it can tell you, you can enter different type of sequences. And these are parameters. So it gives you different type of frames. You can translate using the first frame, the second, the third, or forward. Forward means you're using all the three. And then you can, -1 is the fourth frame, -2 is the fifth frame, and -3 is the sixth frame. Or you can use the reverse 3 frame, and you can use all the 6 frames, okay? And the title is basically optional, and you submit. So I'm going to see if I can submit one of these. Maybe looking for my sequence here. I'm looking for my sequence here. It's my sequence. So I'm going to use all the 6 frame. We'll submit. Okay, so by default, it gives you sequences that are not colored. So to make it easier, you just go and click show all alignment with colors. It's easier to see your stop codon when they're colored. Okay. So I'm going to show here. So you can see from here, this is my stop codon, but this has a methionine and it's the longest. This one, all of them have different stop codons. So basically, this will be my protein. Okay. Okay, I'm going to repeat again. So this is the link. You have the link. You paste your sequence here. And then you have these parameters. You can either select for the first frame, the second, the third, or forward. If you select F, we'll take all the three. -1 is the fourth frame. -2 is the fifth. -3 is the sixth. And you do reverse, we'll select all the three and all the six. And then the codon table always take the standard one, okay? And then you just submit. You can either write your title or you don't have to. When you submit, it will give you the the protein. So why is this important? Because this is a free online tool. When you do your experiment and you submit your sample and you get your sequences or a sequence back, you can translate it into protein. So I'm going to stop here. We have our activity, activity 1. I would like you to go to this website and try to answer the questions. So I'm going to put the questions here for your activity. Okay? So this is your activity here. So you downloaded a file called activity. That file has 5 activities. So the first one is you're going to do translation. So it has a link. If you click on that link, you're going to get that protein. You're going to translate it, and you're going to use all the 6 frames, and you're going to select which is the correct frame. Okay? So type 1 if you're ready to do it. So this is the instruction here.
Steven,
Yes.
maybe you open the activities file so you
Okay.
can indicate what they need to see and what they need to do.
Activities here. Okay, do you see my screen? Type 1. Okay, so let me share my activity screen here. Okay, you see my activity, this activity 1, okay? So you're going to click on this website here. It will open for you, what I just demonstrated, and you're going to use this sequence here. You're going to translate this sequence here using 6 frames, okay? This activity 1, it has a link. And this link will take you to where I demonstrated to you. You're going to use this sequence here. You know, faster, don't take this with it, because faster files start with this. So you can either take the whole thing, or you can also take only the sequences. It doesn't matter. So you're going to copy this and paste and run, and then use all the 6 frames. And I would like you to select out of those 6 frames which one is the correct protein from this DNA. That okay? Okay. So type 1 if you'll be okay. Okay, so that's your activity. I'm going to give you at least 5 minutes to do that. Okay. Someone said link is not opening. The link should. Somebody's saying, are you guys opening the link? Somebody is saying the link is not opening. Is it opening? Type 1. Okay, somebody is, how do you select the correct frame? That's a good question. One is the frame should be at least longer. Two, when you have a stop codon in just somewhere, maybe the first few amino acids, that's not a good frame. The third one is when you have like 2 or more frames that are longer, you can go and do some blasts, or which we're going to talk about later, or you can go to other protein databases and see if you have a domain. But it has to start with the methionine M, basically. So that's how you select your open reading frame. Yeah, exactly. And then frame 4 also, if you do blast, which we're going to talk about in the next few minutes, it has no similar protein in the database. Okay, so I think
Steven,
if you guys are done, type 1 so we can move on.
you had some questions in the Q&A.
Okay, let me go there. Okay, let me go Q&A. Can you send us a document, doc? Okay, this software also says. Somebody asked, can this predict a coding sequence? Coding sequence is basically DNA. This when you're translating from DNA to protein. So there are other tools that are for predicting coding sequences. So the other question here, I think this is it. I think I've answered that now. Okay, so we're going to go back to our presentation, okay? Okay, so now I'll just give I'll stick with my maze example. So now you as a breeder, you went to a field, the whole field was just gone except 1 or 2 maize plant. You sequence those, you got your DNA, you translate into protein. Now what next? So this now brings us what's called sequence analysis, okay? Okay, so we're going to see similarity search by BLAST, secondary structure prediction, CD structure prediction, and multiple sequence alignment. So after getting your sequence, translating them, I want to get your sequences, translating them, what do you do? Because right now, from the video that we watched, the gene was switched on. So you, as a breeder, you are trying to breed your maize. There was a drought. No, there was no rain, but 1 or 2 plants survived. So we were curious, what made these plants survive? Okay, so you got the plant, took it to the lab, extracted DNA. Of course, you have to have also control. And then you send it for sequencing. After sending for sequencing, it came back with DNA. You translated. Now what next? Okay. So this now brings us to what's called similarity search. So why do you want to perform similarity search? One, to find if the related genes in the other organism, that's homologs. So if this may survive, do you have other related genes in other organisms that might help? So it helps identify possible function of gene or protein. So it also can help to predict gene structure, again in cDNA, for example, exon, intron, and also can help predict gene location, okay? So when you do the similarity search, it will help you to do that, okay? So this is how similarity search work. So you have your sequence. This is our maize sequence that made that crop survive in that drought. So what we do, we search this database. So this database has millions and millions of other protein sequences, okay? So after searching the database, what BLAST does, it does pairwise alignment. So then you get your results. So for example, the sequence with the highest similarity to our maize protein was this sequence 28, sequence number 28, followed by sequence number 5, and so forth. So now we will infer that, okay, this sequence number 28 is similar to what we have. That's why our maize would survive. Okay? So how do you search the database? So the most common tool that is used by all the bioinformaticists is called Basic Local Alignment Search Tool, or BLAST. It's widely used and is the most widely cited paper. So it's used for comparing DNA and RNA or proteins. So what it does, it takes a query sequence, the sequence, your query sequence, that your sequence that you are looking for something similar to. It can either be DNA, RNA, or protein. And it finds a local similarity in a large database. So after that, it brings and gives you some scores. So why is BLAST very efficient? Because instead of looking through all, if you get a protein or DNA and it aligns, it doesn't align from the start to the end. It takes a bit of chunk and it's called local. That's why it's very, very efficient. Okay. So there are different types of BLAST. One is called BLAST N. So if you have a DNA and you want to compare DNA, you use what's called BLAST N. You get your DNA sequence, you query, and the database has DNA sequences. And BLAST P, that's what we're going to use through this webinar. You take your protein sequence, and the database has protein sequences. And BLAST X, you take your DNA, and it translates it into 6 frames. And then now it goes and use all those 6 frames to look for the protein. And that is very, very slow, really very, very slow. And T+N takes your protein and then compares it to nucleotide database and so forth. But the most common BLAST method that I use are two: BLAST-N for comparing nucleotide and BLAST-P for comparing proteins. Okay. So how do you use BLAST? So there are 2 ways that you can use BLAST, by the way. You can download it, install it on your computer, and run it on your own local computer. That's a little bit tricky. You have to know at least Linux to do that, or you have somebody to help you do that. And secondly, you need a very big storage. But the easiest way is to use the online tool. So this is the address. when you go there, you'll find different types of blasts. You can either choose blast N when you're doing DNA or RNA, or blast P. You're going to use blast P. And then after that, you paste your sequence and so forth, okay? So it also has very different databases, but the most common one is called non-redundant database. And I always tell people use non-redundant databases. You can select different type, but I always tell people use non-redundant databases. And then you can also filter by organism. You can take humans, you can take, and then it gives you what they call expect E value, which is like the P value. And that is basically 0.05. And then you click blast, it will give you results. Okay. Okay. So when you go to that web page that I showed you, this is what you get. You get nucleotide blast, blast X, T blast N, and protein blast. So the most common ones are protein blast and blast N. For us, we're going to use protein blast. Okay. Okay. So when you click on that, you get a window like this. You get a window like this, and then it will ask you to either enter session number or you put your first file format. You can also choose your file, or you can, if you want to read more about the guide, you can just click more, but we are not going to do that. So for us, we're going to use protein blast because after translation, that's what we're going to use. And then the database always non-redundant. And then the blast P is your the algorithm. Okay. We have to have blasts, but we are not going to use that in this webinar. So let me talk briefly about blast. So sometimes when you submit your protein sequence in blast and you don't get any results, you can use blast. But the difference between blast P and blast is that when you're searching using blast P, it goes in the database and compare your query with each protein in the database. But blast, what it does, after getting all the similar protein, it creates a multiple alignment and uses a multiple alignment now to search. And that's more sensitive. So sometimes people use blast, but we're not going to use it here. Okay. So I just wanted to tell the difference between blast P and blast. Okay. So the query, you can either put faster format and you can also use a session number. So a session number basically is the ID of your sequence. And then the parameters. Always, when you start searching databases, I recommend to use default. So this is a parameter of blast. The maximum sequence target, that means that the sequence that you'll see is 100. You can change this. And the expect threshold is 0.05, which is the E value. And this is the word size. That's what blast uses to search. And the scoring matrix is blossom and so forth. So when you're using blast, always just leave everything like this, which is default, okay? Unless otherwise, just leave, because by the time you build algorithms, select the default, that means they've done rigorous work. So always leave it at default. So for us, what we're going to do for activity, we're not going to do anything with the parameters, okay? After that, then you blast it. Basically, blast means search. and then it will go into that database and searches. It comes back with something like this, blast statistics. It will give you the E value, which is 0.05, but this is an E value. It gives you the identity percent. That means the identity means the amino acids that line with your query and that one in databases and so forth. Okay, for you to decide whether that, your sequence is close to what's in the database. Okay, so let me go back and repeat this again. So why do we do similarity search? We do similarity search to find protein or DNA in the database that are similar to what we have sequenced. I'll give you an example of the drought tolerant maize. So we found a protein that made that maize to be tolerant to the heat because the rest died. So there's a tool called basic local alignment search tool called BLAST. You can install it on your computer, but you need a big space and it slows your computer. But you can also run it on a web page at NCBI, and this is the address. So there are different types of BLAST, but the most common ones are for BLAST-N for DNA and RNA, and BLAST-P for protein. So for this webinar, we're going to use BLAST-P because we already translated our protein. Okay. So to use BLAST, you need either your sequence in a faster form or you can use a session number. But if you just if you just add your sequence being from the machine, that means you don't have a session number yet. So you just use the faster format. So when you go to that web page, this is what you'll see. We give you different type of blast. The most common ones are nucleotide blast and protein blast. For us, we're going to use protein blast. If you click on this protein blast, give you this window. And in this window, if you want more information, you can create more. And this window here, there's a place where you can paste your sequence. And this is the window, okay? And then you can choose your database, but I recommend that you use non-redundant database, okay? You can also select your organism, okay? And then you can also go to parameters. I also recommend you leave the parameter as default. And the max target sequence is the number of results which you get. And the expect value is basically like the p-values that will tell you how statistical significant your search with that of the protein that you've got from the database. And then the matrix is scoring is blossom 62 and so forth. Okay. Then after you get your results, it will give you the E values, it will give you the identity of the alignment and so forth. Okay. So I'm going to show you what I did, and then I'm going to show you the practical part. So I did 2 blast searches. The first one, I used this protein here. So this was my protein, okay? It's blast P, and then I just did the blast without changing any parameter. The parameters, I didn't change anything. That's why I put here, do not change. So when it's running, this is what you see. This is the screen you see when it's running. That means it's searching the database. You have to, first of all, have to make sure that you're connected to the internet, otherwise it won't work. Because it's being run in the US, okay? It's because NCBI is based in the US. Okay, so this blast search is searching. These are my results. So you get something like this. So it gives you 100, okay? So these are the results. So it first gives you a cluster. This is the cluster, and these are my results here. So look at the E value. This is very, very high because I need 0. At least less than or equal to 0.05. So that means the protein that I'm searching is not there in this database, okay? And this is the session number. That's one way of looking at it. You get the results, look at the clusters here, look at here, come to the e-value first. So this is very high. That means I cannot trust this because sometimes you don't have this protein, maybe it's something novel, okay? Okay, then next, I go to the graphic summary. So the graphic summary also gives you scores, alignment scores, how they align. So anything less than 40 is not good. So it will be black. For a good sequence, should be greater than or equal to 200. It will be red. So this will also guide you. So mine is black. That means the alignment wasn't good. Okay. And then the alignment, look, this is the only section that aligned. You see that? The only section that aligned was this one here. That's why the expect value was so low. You see that? And then, of course, the taxonomy. So I didn't trust, basically, this results statistically is not significant. So then I did the blast search, the second one. So I did the same thing, put the second sequence here. I didn't change any parameters. So now if you look at this, my e-value is very low. See this? E-value is very, very low. That means significant. See the cluster. Now when I look at the graphic here, you see this, my graph, the alignment score is very, very high, greater than or equal to 200. So I'm very, very at least confident that my protein now has a similar protein in the database, okay? Look at alignment, almost perfect alignment. Look at the query with this. The identity 100%. That means my protein aligns perfectly with this protein in the database, okay? And then I have such statistics. So when you do BLAST, you have to report this. And how do you get BLAST statistics? You go to search summary. Because if you're reporting in any manuscript and you don't have this, they will reject your manuscript. Why? Because every search, the eval depends on the number of sequences in the database. This is 461 million. So if you do the search, you have to report BLAST statistics. And this is very important. You have to put your number of sequences in the database is 400 million and the date that you did that, because a day after tomorrow, this number will change because people keep putting their sequences there. So after blasting, you have to make sure that you save this. When you're doing blast, it's like you're doing an experiment, by the way. And the blast piece statistics is very, very important. Okay. Okay, so I'm going to go and do one site with you, and then we'll go to activity 2. Okay. Let me go and see if I have some questions here before we go there. Somebody asked that I'm purely statistic background with no biology knowledge. Can anyone recommend areas to read and understand the basic, please? Let me do some research. I will get back to you on that. Okay? There are some books that are for people like you. Okay, I think we're okay with that. So let me go and go to blast search here. And then I do one, and then we'll do that together. So you see my screen, type 1. Okay. So this is the NCBI database. Okay. So you have nucleotide blast and protein blast. So I'm going to go to protein blast. Okay. So if I click on protein blast, this one will turn blue. That's what I need. So I'm going to look for my sequence here. No, I'm going to do the second one. It's better. First one. Okay, so I'm going to use non-redundant database, blast P. The parameters, I'm going to leave them the way they are. These are my parameters, okay? I'm going to leave them the way they are, and I'm going to blast. So see, it's searching now. Okay. What did it say? Warning, one or more characters replaced by X. Something happens here. No significant, no significant, no significant similarity found. That's interesting. Let me go back. Hmm. Let me go back. Go back to NCBI. Okay. Go back and try again. Plus P. That's my first format. 3 factor. Sometimes when you get files from Microsoft Word, sometimes these things get corrupted. So I want to make sure it's not corrupted because this right in the beginning. Let's try again. Okay, this makes sense. Okay, so these are my results. So this is my E value. So this is a class that's my E value, and I wanted 100. So this all these are what are similar to my protein. So the top is this one here. So this is what we can do. If you see this accession number here, if I click on this accession number, it will take me to the protein itself. So this is now, it will tell me the organism, the definition, and so forth. Okay. If I go back, if I go to look at the graphic, because my E is very low, look at the graphic, you can see here it's all a red. So greater than or equal to 2000, that's good. Look at the alignment. The identities is 100%. So this is good. So that means the protein I have is similar to this protein here. Go to taxonomy, so give me this organism here. Okay. So this is how to use blast P. Let me repeat again, then we'll go to activity, okay? So NCBI. Let's start here. So if I click on this, it will bring me here. And I'm blast P, protein blast. Then I'll paste my sequence. Everything will be, let me make sure that there's no gap here. There's no gap. No gap. No gap. Okay, my parameters are just going to be default. And then I'll just blast. It's running, searching. Okay, these are the results. The clusters. And then I need to see my search summary. This one. Search summary will give me my summary statistics. This is number of sequences. So this was, the database was updated in August 19th. They haven't updated it yet. So this will be, when I'm reporting this, I have to give the date when the database was updated, the number of sequences in the database, and so forth. Okay, so we're going to go and do activity 2. I'm going to open, uh, I'm going to open activities. Okay, you see my screen? If you can see my screen, type 1. Where the activities are. Okay, so this is the link. You're going to click on this link. After opening the link, you're going to copy this sequence and do the blast search, okay? This is your link for activity 2. You click on the link, it will take you. If I click on that, it will bring you here. And you select protein blast, and will give you this window here, and then you paste your sequence here, and then you blast, okay? Can we do that? In the meantime, I'll go and answer questions from the Q&A. Okay, let's do activity 2, okay? Q&A. Okay, let me see. Q&A. Okay, somebody asked me that when you select the code, when we're translating, so what are the other function, the drop box, when it drop down? So sometimes, especially when you're translating uh, bacteria or fungi, sometimes they have different genetic code. They're kind of a little bit different. Then you select those ones. But most of the time, people use standard one. So sometimes it's good to try different types, but I recommend the standard one. Okay. Let me get another one here. Okay, the question, what sequence are you using, DNA or RNA? We are using DNA. Okay. So what's the name of the website? So this is NCBI. That's the name of the website for doing blast. It's NCBI. Okay. Okay, somebody asked me, why using DNA rather than RNA since it's RNA that is translated? Okay, so RNA gets information for DNA. That's why we're using DNA. So RNA is just a messenger. It gets that information from the DNA, from the DNA. Okay? Somebody, what happens if there are no star to indicate stop codon? So that means there was no stop codon in that. So what you do is if it starts with methionine, then that's your protein. But if not, then You can go do BLAST to find if you have any similar protein. How do we ensure data integrity when doing our work? How do we ensure data integrity when doing your work? Of course, you have to follow the protocol of what peace taught, how to protect your data. And BLAST is really safe though. I mean, they have NCBI, they have very good data security. Don't give colors in my side. Or somebody said disalignment with colors, don't give colors in my side. Okay, so when you this is taking us to translation. When you translate, on the lower side, there's somewhere that shows you just click so that you can get colors. Unless you click that one, you will not get colors. Let's see here. Yes, you can select all 6 frames in one time. Somebody asked, can I select all six frames in? Yes, you can select all the 6 frames in one time. You can do that. Okay. Have we done a blast search? Type 1 when you're done. And type 1 when you're done your blast search. So was it significant? What was the E value? Okay. Let me see here. So what's the session number of the top sequence? So you see my slides here. These are the questions. You perform the blast search. You report the E value, provide the session number of the top matching sequence, and state the number of sequences available in the database. So the session number is easy because it's on the right of your first sequence. But to get the statistics, you have to go somewhere and click so that you can get the statistics. Okay? Let's see here if I can. Okay. So I think we can move on. Since we have done our blast, let me go back to my slides. Okay. So we have covered our translation to amino acids, to protein, and we have search. Well, the next we are going to, the next level is secondary structure. Because sometimes the amino acid is a primary structure. Sometimes the secondary structure is much better. So what's the secondary structure? This is a local folding pattern of the polypeptide, and they're stabilized with hydrogen bond. To that, you have the 3 types. And each type depends on amino acids. So you have alpha helix. These are cold structure and they're stabilized by intrachain hydrogen bond. They have beta sheets. These extend by hydrogen bond, but they can be parallel or antiparallel. And you have loops that connects the alpha helix and the beta sheets. So these are the secondary structures. There are the 3 types. Okay. So this is just a cartoon for alpha helix. So these are mainly the amino acids that are composed of alpha helix. And these are amino acids that are composed of beta sheets. So you have this alanine, leucine, methionine, glutamine, and so forth. Okay. So what's the application of this secondary structure? So they help in genomics annotation. So when you have this secondary structure and you don't know, like you don't know which protein, you did blood search and you can't find. If you do the structural analysis, you can find the structure of protein that's similar to your protein. And they can also support genomics and laboratory studies. Okay. So they help in genomics and annotation because sometimes blast fails to get your protein. Because sometimes you have these proteins at the primary structure, that is at amino acid level, they may not be similar, but if you go to the next level, secondary structure, then they become similar. Okay. Another important function of secondary structure is structural biology. So when you do structural biology, people do what's called homology modeling. So when you're trying to predict 3D structure, you do what's called homology modeling. So it provides a template for 3D modeling. And then people will do protein engineering. So when doing protein engineering, when you have these secondary structures, it helps you to modify those functional domain and also help to design in synthetic biology. So primary structures are okay, but they have their limitation. That's why you go to secondary structures. Okay. They also help in drug discovery. So because drugs always act on protein, but when you are designing drugs, at least they help when you have secondary structure to help you predict the mutations where the disease or protein are related. And also it also helps in disease research, like example, interpreting genetic variants or neurodegeneration or antimicrobial resistance. So if, for example, you have antibiotics that bacteria is no longer responding to that can be caused by disruption by mutation. So if there's a mutation in amino acids, if that was an alpha helix, that might change because those structures are based on amino acids. So if there's a mutation, the structure will change, then maybe the bacteria can become more resistant. And also in biotechnology and also vaccines, so it helps in designing when you're dealing in biotechnology. So it helps in performing therapeutics and protein enzymes. So it's very, very important, this structure, secondary structure prediction. Okay, how do you do secondary structure prediction? Okay, there are very, very many um, methods out there for doing that. You can also, it's like BLAST, you can download and put on your computer. But I selected this one here. It's a server based in UK because it has different methods of the prediction. So again, it uses faster format, and then you can use your email, and then you just paste your sequence, and then you submit. Then after submitting, you get your results. Okay. So these are the outputs. It gives you your alpha helix, your beta sheets, and the coil. It also give you confidence score, and you can also visualize this graphically. So you can view the result online, or you can also download. That's why I selected this to share with you. So this is the website. It's called JPredForm. Okay. So what you do, if you go to that website, it gives you information about the home, other information. It also gives you even a tutorial. And then what you do, you just paste your sequence in the window like this, and you make your prediction. And then again, I always tell make everything default, don't change this parameter. Okay. Then when you submit, when the job is running, you get a window like this. Okay. Then these are the results. So on top, you have the sequence that is submitted. That's your primary sequence, which column in the amino acid. And then you have your prediction. The red are your alpha helices, and the brown are the coils, and the greens are your beta sheets. And then it gives you different algorithms of which how they predicted. And then you can also view results HTML and also in others. Okay. So this is why I like this platform. So it compares different methods, and then for you to accept the results, for example, this one JHM predicted this to be beta sheets, but these two predicted to be alpha helices. So this one, you take it a grain of salt. But if you look at this, these three of them predicted the same to be alpha helix, and this one predicted to be these three predicted to be what? Beta sheets and so forth. So this method is good because you have this. It uses different algorithms to compare, and then it gives you all the information also about what this all means. Okay. So I'm going to run with you, and then we'll do the activity together. But before I do that, I'm going to go back and talk about the website. Okay. So it is based in UK. It's called JPredFo. When you go to the website, you can read more about what it does, home, about, and the rest. And then there's a window where you paste your sequence, and then you make prediction. Okay. So it also all these prediction method, they have their parameters, but always leave them as default. Okay. So for making prediction, submitting, you find this is when the job is still running, and then gives you this information here. The alpha helix is red, the beta sheets are green, and then the coils kind of like brownish. Okay. And then when you go further down, it gives you all this information. Okay. So I'm going to go to the website. So this is the website. Okay. So when you go to the website, it comes with a sequence just to guide you. But I'm going to get rid of this sequence here. Okay. So I'm going to use my own sequence. That's my sequence here. Prediction. So it's running. So give you the time. So also might take longer, depends on how fast your computer is. So this is taking longer. It depends on how fast the computer is and also how when there are many people in the queue, what's your position? So sometimes you have to wait. So it's still running. So that's why they ask you if the job's taking too long and you have already provided an email, feel free to close the browser window. But I didn't provide my email address. So if I close the window, it will be gone. So just have to wait for a little bit. Wow. Okay, meanwhile I'm waiting. I'm going to so that also you can get started, okay? So I'm going to go to activities. I'm going to go to your activities, okay? So if you open your activity 3, see this activity 3, the one you downloaded from the Google Drive. If you click on this link here, it will take you to the server where I went and it's running. If you open this link, you copy this sequence. Start from here. Don't copy that session number. Just copy the sequence by itself. Start from M. And then also run. Okay? So this is your activity 3. Go to, click on that. Click on this link. It will open for you. And copy from here and run it. Okay? So somebody said, I've tried the JPEG using the link in the activity and I'm getting software error. So what does the error tells you? Does it give you anything? Patricia, you said you're getting error. What does the error say? Are you guys, is everybody getting an error or is you trying to, is running? It's running type 1, activity 3. Okay. Answer this question. So as somebody asked, what's the essence of translating? Can we blast the sequence product directly? You can use T-blast, but that is very, very slow. Why don't you translate and then just do protein blast instead of submitting your sequence and then translating to 6-frame? That will take time. It takes a lot of time. Okay? You could do it, but it takes time. Let me see the question here. So someone's asked, can you have an idea about the cost of DNA sequencing? The cost depends on one, on the machine, two, on the company, because if you're using Illumina, it's different from if you're using Oxford Nanopower. So that one depends on the company. Do you have laboratories performing sequencing in Africa? Yes, we have some. We have laboratories in Africa doing sequencing, yeah. Which blast, which blast is most powerful? That's a good question. if you compare BLAST P, I'm just going for protein. BLAST P and CyBLAST is more powerful than BLAST P. Why? Because CyBLAST uses alignment for search. BLAST P uses just pairwise BLAST. So BLAST P, when you submit your sequence, the sequence goes to the database and it goes to do pairwise alignment. It's going to compare each sequence in the database by itself. But with CyBLAST, after doing the first search, it's going to get the results. And then it's going to do alignment, aligning all those sequences, and use that alignment now to search. That's more sensitive. It's sensitive compared to BLAST P. But I cannot compare BLAST N and BLAST P because I compare apple and orange. Because BLAST N is searching for nucleotide, BLAST P is searching for protein. But when you're using CyBLAST and BLAST P, you're searching for an orange and an orange. That's the, yeah. Somebody say, how do you have a sequence? There are very many ways of getting a sequence. One, you can get your sample. For getting your sample, you send it to a sequencer company, and then you get your sequence. Secondly, you can go and get a sequence from a database. It depends on what you want to do with that sequence. So those are the ways you can get a sequence from. So I'm using NCBI Blast, but I have a session number. So somebody says, so if I'm using NCBI Blast, but I have an accession number of the organism from another database, how can I then convert the database into NCBI database accession number? Okay, so there are three databases that you can use in this. I mean, there's one in Europe, and there's one in Japan, and then there's NCBI. So all those three, they collaborate. You can send your sequence in any of that. So if NCBI has an accession number, that accession number will be at the same in all those the other two. So the accession number you have in NCBI can also work in other databases. Okay. So another person asks, how do you know which score metrics to select? Okay, so I didn't want to go to score metrics, but I'm going to go there since you asked. So the scoring matrix, we have BLASM 62. That is the most commonly used, this standard, because that one is basically it's in the middle. But if if you have a protein that has very low similarity, you have to use either BLASM, a lower BLASM, or you have to use a PAM, a higher PAM. So it depends on your protein or your amino acid. Okay. So let me answer. Let me go to another question here. Blast the frequency product that I got. Can we blast? If you have DNA, you can blast using DNA, blast N. But if you want to, if you have protein, it's advisable to use blast P. So let me have another question here. So I'm answering questions in. So the question, can you show again how to get the statistics summary? Okay, I'm gonna go back and show how to get statistics summary. Okay, somebody asked that. Okay, so let me go back to blast. I'm gonna go back to protein blast, okay? Somebody asked how do you get statistics summary? I'm gonna use this to do blast. Okay, I'm gonna use that. So this is my blast P. Okay, so the protein blast P, let me blast. So it's running. Okay, so to get your statistic, you see this search summary? Yeah, that's where you get your summary statistics, search summary. Okay. Okay, so go to search summary here. That's when you get your search statistics. Okay, go back. Answer some questions here again. Already answered that. Okay, somebody asked, I didn't understand why you chose the first OR of open reading frame. Okay, so let me go back again to that. Okay, let me go back again to that. Okay. So we're going to translate all the six frames, okay? Okay, let's run this. Okay, so to make it easy, somebody asked about coloring. So by default, you get this non-colored. Okay, by default, you get this non-colored by default. Okay, but to make it easy, you go to this one here. Easier that way. You click on that. So the question: why did we select the first open reading frame? Okay, so to select your reading frame, the first it has to be longer so that you don't have the stop codon. And then secondly, most proteins start with methionine M. So let's start with this one. This one starts with methionine M, and then the stop codon is here. So from methionine up to here, this is a very long, the length is very long, okay? Okay, let's take the second frame. The stop codon just in the beginning. So that alone was just qualifies it. Another stop codon is here. Another stop codon is here. The stop codon just makes you stop making protein. If you go to the third frame, the stop codon is here. So this is very short protein. To go to the fourth frame, the fourth codon, the stop codon is here. Go to the fifth frame. This is a little bit longer, but still, the fourth codon is here. If we go to the sixth, again, so we selected open reading frame number 1 because, first of all, it has a methionine in the beginning, and then it's the longest here. Okay? Okay, so I think that should answer that question. So let me go back and see what some more questions. So we're going to stop here today, but I need to answer more questions. Then we'll tell you the assignment for tomorrow, for the take home assignment. But I need to answer some of this question here. So what's the question? What's the purpose of using non-redundant database? Non-redundant means the database has been basically worked on very well. It has been curated so that at least when you use it, the sequences that are there, at least they've done some good work on that. So that's why it's always good to use non-redundant database. So where in Africa? So where is Africa? Okay, so Becker, which is, someone's asking about the sequencing. I think Makeri should also have some sequencing machines, if I'm not mistaken. But I know International Livestock Research Institute have sequencing machines. Uh, some some research centers have sequencing machines. So I think there's also one genomic center in West Africa that have sequencing machines. Okay. Okay. So somebody asked, which database commonly used to prepare for BLAST? So BLAST always it depends on BLAST is a tool. So when you're using NCBI, the database is NCBI. When you're using like SwissProt, the database is SwissProt. If you download BLAST on your computer, your database will be your computer. Okay? Okay. The last activity, how to translate it to show colors and so I already done this. Okay. How can you share the material share today? We'll share the materials for today. We'll do that. Okay, so let me see. How primer sequence forward and reverse? How can I blast? Primer sequence. This is a good one. Primer sequence forward and reverse. Can you blast them separately? Are they I think you could blast them separately. So we have only 4 minutes left. So I'm going to spend time now about the homework, the assignment. So you downloaded another file, which is called, I think, metagenomic packages. So what would like you to do when you go home, try to install those packages. Let me see if I can go there. Okay, you see my screen? Type 1. Okay. You see this, you downloaded this one here. These packages, we need these packages tomorrow. Tomorrow we'll finish today's webinar and start metagenomics. So when you go home tonight, try to install these packages. Just try to run this script. You should be able to install all those packages, and we're going to use them in metagenomics. Okay? Any questions? Let me go back here. So we said I failed to read the BLAST query. What do you mean failed to read the BLAST query? Did the BLAST work, Renee? I failed to read the BLAST query. Did the BLAST work, or you failed to read the results? Some more question. So I think we have run out of time. We have only 2 minutes left. I can answer some few questions.
Yes, you can answer questions before we close.
Okay. Let's see here. Has the link been shared? So we don't, somebody has the register link been shared? I think that was shared before. Can you show more light on the interpretation of summary set? Okay, so somebody want me to show light on interpretation of summary statistics. Let me go back there. That should be the last one here. So let me go back if I can see that. It's my blast. run BLAST again though. Okay, we'll go back and get a sequence here. Okay, this is a summary. Okay. So let me explain this. So the program is BLAST P. What size is 5? So remember I mentioned that BLAST, when it does search, it doesn't go, it's not global. That means it's not going to start from the first sequence to the last. So it takes five amino acids in chunk. That's why that's the word size. And then 0.05 is the expect value. This size is the size of the results, the 100 sequences that you need. Okay, gap cost. So when BLAST is doing some search, there's what it gives. It gives penalty. So the opening. So if it starts searching, the penalty that gives there's one for opening and there's one for closing. And the BLASTM 62 is basically the matrix. And then there's filtering, the genetic code, I think this is one, and the window, I think is windows, I think is the size of the size of the sequence that's supposed to go through. And then the database, because every now and then the database is updated. So the last time this NCBI database was updated was August 14th. That's why when you're reporting your BLAST search, you have to give this. And then the number of letters. So this is the number of amino acids in the database is 147 billion. And the number of sequences, 461 million. And then inverse query none, we didn't use that. So these are basically statistics that are used for calculating the e-values. There's lambda, there's k, there's age, the alpha, all these are involved in statistics of how you calculate the e-values, basically how to interpret this. But when you are reporting, you just have to give them all of this one here, especially the date, the number of sequences, and then some of this. I hope this helped. Thank you. I think we've run out of time.
Yes, we have run out of time, colleagues. I think for more questions, I think you are in our WhatsApp groups. You can always continue posting questions and others will respond to you on such questions. But I also encourage all of us to go to our Ruforum Network YouTube channel to revisit the training of today. And I know the link of the training materials have been posted now and then in the chat. So let's close for today and we meet again tomorrow at the same time. Have a good evening, good afternoon, good night to everyone. See you again tomorrow. Bye.
Bye.